Welcome everybody to our last meeting of April 2022. Time is passing quickly. I appreciate you for coming. Appreciate our speakers for being willing to talk to us today. As always, I need to remind you that this meeting is live streamed and recorded. So please keep that in mind. We have two speakers today lined up on two interesting topics. We have Lucas coming to give us a second talk. I remind you, you're all welcome to give multiple talks, so we appreciate that. As always, Reinhard and myself, Jim Sluka and Lorenzo are available for suggestions, comments, critiques, and we're always willing to do better if we can. We have our various communication channels. I don't know how effective they are sometimes, but please help us use them. We've been trying to do a little bit of updating on the IMAG MSM wiki page. In particular, if you have articles that you want to post there that are related to the working group, please send those to us and we'll put them up. If you have conferences and those sorts of things, please send them to us. If you have job ads, you have postdoc positions to fill, faculty positions to fill, those could be distributed to the working group as well. So please do take advantage of that. As always, I remind you about our YouTube channel. Please help us get the word out on our growing archive of talks. I'm trying to do a little bit more indexing there. People who are willing to help index would be much appreciated. We did create a Twitter account called MSM Viral. If you use the tag MSM Viral, that would help distribute things in a consistent way. Contact Jim Sluka if you need any more information about that. Are there any announcements before we get to the meeting proper? We have a steering committee meeting tomorrow at 11 a.m. Eastern Daylight Time. If you think you should be invited and you haven't received an invitation, contact one of us, Reinhardt, myself, Lorenzo, or Jim to send you the invitation. Any other announcements before we get started? Besides the fact that Reinhardt's model sharing paper is accepted for publication. So that's a nice thing.
It's building an immune digital twin.
Yes.
I have a recent paper or recent editorial accepted too on, it's one of the specialty grant challenges for frontier systems biology. Incorporates a lot of the digital twin stuff we've talked about along with other things too.
Right.
Great. Well, if you want to put the links to things in the chat and definitely we can announce those. If once you have the links, we can put them up in the slides next time and also put them up on the website. That's great. Any other announcements? Okay, coming up on next week, we have Hayden Fennell from Indiana University, who's going to be talking about use of models in education. This is a little bit of a different topic from our usual, but we've heard a lot over the past 2 years about how models can be used to convey information, to influence policy, to communicate. And so we thought it might be interesting to hear from a specialist in education-related scientific modeling about approaches to using modeling in education and communication. So we have a second slot available if anyone else would like to talk. Joy has agreed to speak on the 12th. We're very grateful to her. And we have a talk on May 19th, Chang Zhang from Emory as well. I hope to get the titles for those soon. We have slots available on those days or going forward. As always, please suggest people you'd like to hear, yourselves, your students, your postdocs, and help us come up with an interesting roster of people that you want to hear. So I won't take any more time from our speakers. Our first speaker today will be Lucas, who will be talking about how neural networks learn to control dynamical systems. We've had a number of talks on control theory over the past 2 years, and so we're looking forward to hearing this one. I'll turn it over to you.
Okay, thanks. Something else came to my mind when you mentioned additional announcements, and there's one interesting conference that I also advertised a bit in other meetings, and some colleagues in Louise. They're organizing the Ising lectures for, I think, about more than 20 years already, and so for 24 years already. And it's a conference on phase transitions and critical phenomena, but there are also a few lectures on social dynamics and disease dynamics. And it's an online conference, and if you're interested in attending that workshop or conference, feel free to do so. You can just Google Ising lectures and you will find the corresponding links. And now I need to switch to another computer, and I hope that you can see my screen, because I have a few animations that don't run on my laptop. I will be talking about controlling dynamical systems with artificial neural nets, and it's a project that we started in 2019 because we were interested in controlling large-scale dynamical systems that we previously used in disease modeling. And I started talking about that with Nino Antula-Fantolin, who's still affiliated with ETH. And then later on, we involved Thomas Lasikis, who was a PhD student at ETH back then. He now completed his PhD a few weeks ago. And then a few months later, COVID hit, and we then realized that it's kind of a useful project, and we invested a bit more time into that, because it turned out that artificial neural nets, or basically neural ODEs, can be used to very efficiently control large-scale dynamical systems. And before focusing on the actual algorithm, I will give you some background information on traditional control theory and a few examples that are commonly used in the control literature and in the reinforcement learning literature as some standard examples or standard baselines that one should be able to control. And then I focus on the neural network-based control approaches that we develop, and I show you a few applications. And I also wanted to basically, with this presentation, ask for some input with respect to possible collaborations on topics that are related to, for example, what I had in mind, the treatment protocol optimization, which can be quite challenging, and I show you a few examples. So I think there are also some very interesting applications with respect to computational medicine or computational biology. Okay, but I start with a few standard examples that are commonly used in the control literature. So one example is the lunar lander, and the goal is to, you've all seen that, basically make sure that some spaceship or lunar lander lands in the specified area. And that's a typical example that one can solve with RL-based methods, or one can also try to derive some analytical results for that. And then there are, of course, some real-world counterparts like SpaceX that are basically using similar ideas. And another standard example is the famous card poll. Like, these are all small-scale examples, right? There are a few variables that one has to optimize. And if you read some papers also in the machine learning literature on controlling dynamical systems, it's basically always card poll, lunar lander, some quadcopters, and And sometimes in applications, you also see these robot arms, which is an example that I took from the Bear group at UC Berkeley. And they use reinforcement learning to train robots or robot arms on different tasks. And now, like if we think of traditional control theory approaches, we basically call a system, a dynamical system, controllable. It can be steered from any initial state, x naught, to any target state, x star, in a finite time. So that's a simple example of time-dependent control. And usually the goal is to minimize some cost functional, and there's usually some integrated costs that could be, for example, something like the number of total infections in the population. So if we integrate overall infection events, and there's also some final costs that one can incorporate in this cost functional. And the constraint is that there is a certain dynamical system, or that the system state obeys a certain dynamics. And now there are different approaches in the traditional control theory literature that one can use to solve these types of problems. A necessary condition for an optimal solution is provided by Pontryagin maximum principle that has been derived by Lev Pontryagin in the I think in the 50s last century, or in the 50s or 60s of the last century. And there's also a formulation that is based on the Hamilton-Jacobi-Bellman equation, or the HJB equation, which is necessary and sufficient condition for an optimal control solution. If you really try to apply these methods to general control tasks, like the math and also like solutions can become very complex, and for some tasks it's not even possible to, or for many control tasks it's not even possible to derive analytical solutions. And then there are different numerical methods that one can use, but they are also, depending on the type of control problem that one is studying, can be also very challenging to implement. And we also compared some of our methods with numerical optimal control solvers, and there are huge performance differences, and I will comment on that a bit later. Now the question is, how can we control, say, high dimensional and also potentially stochastic dynamical systems? And what we use is the framework of neural ordinary differential equations, and that's kind of new in the ML community. So there were a few papers in the past 2 or 3 years, although some of the ideas are also mentioned in earlier papers, I guess. And the basic idea is that one starts with a certain dynamical system. So it could be a mechanical system like coupled oscillators, or a cellular dynamics, or we could also think of social dynamics. And to model these systems, I'm now mainly thinking of some network dynamics. So we have some type of adjacency matrix that's describing connections between different individuals or nodes in a system. And then we use some kind of numerical solver. It doesn't really matter which numerical solver we use. We can combine different types of solvers with these neural ODE controllers. And we just time unfold the dynamical system, and we integrate it numerically. And before controlling the system, we end up with uncontrolled ODE dynamics. And in the second step, and that's also connected to traditional control theory and structural controllability, we identify certain nodes as nodes that we wish to control. Could be also so-called driver nodes. But in general, we just select a few nodes that we wish to control, and we use some matrix B that is describing which nodes we connect to which controller. And the control function that we use is, in this case, so there's no feedback control. We also studied feedback control systems, but in this case, there's just a time input. So time is the input variable of the neural network that we use to represent a control function. And the output could be one-dimensional, it could just be one scalar control output, or it could also be that we use different control functions that we connect to different nodes. And again, like the control function is just parameterized by some artificial neural network. And because of universal approximation characteristics of many of these artificial neural nets, that means if we use the right basis functions or the right activation functions, we can basically approximate every function that we wish to approximate. And so we just do that in the context of control. And then we solve our control problem. And in the first few iterations, we may not end up in the desired target state, but then we just calculate some difference between reach state and target state, and we backpropagate gradients, update neural network weights, and train the neural network to learn the desired control function. And that's the corresponding schematic. So we have a time unfolded ODE system. We calculate the state evolution. We calculate the corresponding loss function J. We backpropagate gradients. We adjust neural network weights, and then we hope that we really learn the desired control function. And that's also something that has been studied a lot in the control literature, and there are many applications, of course, also in engineering. But what's interesting is, and I now show you a simple example, linear dynamics. What's interesting is that we can learn near optimal control solutions without really specifying that we want to learn these optimal solutions. So there's some kind of implicit regularization or some kind of implicit retrieval of these optimal control solutions that one can observe in many systems that we studied. So we start with some linear dynamics, and we just control one node or one part of the two-dimensional system, and we start at a certain point in the phase space x naught, and we want to steer the dynamical system towards x star. And what you see here, like all these different solutions correspond to different neural network outputs for different numbers of training epochs. And the dashed black line is the optimal control solution. And now you may not have seen that on the previous slide, but we are not really optimizing for optimal control. We're just minimizing the distance between reach state and target state. So there's no implicit regularization with respect to what's called the control energy, or the integrated cost over U squared. That's some kind of like energy, or like some kind of energy equivalent, right? Or some type of integrated cost that one often wishes to minimize in optimal control. And we didn't specify that, but still we can observe that the neural network basically learns to approximate optimal control solutions. And that's one example where we have observed this type of behavior, and that's the corresponding animation. And you see that for like after about 2000 training epochs, I guess, the neural network learns to approximate the optimal solution, and that's the learning process that I'm visualizing here. Right now, the energy goes up, but we also get closer to the target state. And that's still very low dimensional. So it's just two nodes. But we can also go to larger. We can also go to larger systems. For example, oscillator dynamics, Kuramoto dynamics. And these are oscillators that we arranged on different types of structures, on lattice structures or other types of networks. And then we study the phase evolution in an uncontrolled system. If we are below a certain critical coupling strength in these in these oscillator settings, we observe that the oscillator phases basically diverge. So there's no synchronization, but we can define different types of loss functions. One loss function that tells the neural network that we want to separate these oscillators in two separate phases. So we end up with two separate phases on the lattice. And we can also tell the neural network that we want to synchronize these oscillators. And it can learn the corresponding control signals relatively fast. And we compared it also to other numerical optimal control solvers, so-called adjoint gradient methods. And these methods often have the problem that they implicitly assume some, or they don't implicitly assume, you basically explicitly add the energy regularization term in these frameworks, and there are too many functions that are coupled. There's a gradient descent in the control function. You have the evolution of the system states, and then there's also some coupling to the adjoint variables that you need to solve for, and that can lead to stiffness problems. And it's usually very hard to solve, while in the examples that we were studying with the direct neural network solver, you don't have these issues, and the optimization is typically very fast and efficient. And we also applied it to the seed dynamics on a lattice, and we compared it to some very recent reinforcement learning methods, so some state-of-the-art RL techniques, and we also compared it to other baselines. And what we observed is that because we're, unlike in RL, we're accounting for the underlying dynamics directly in the optimization process when we use these types of neural network optimizers that we developed, or that we applied to these problems. And as I said, like unlike RL, there is some gain in efficiency because we use that knowledge on the dynamics. And it's not only efficiency, but we have also seen, like in this example, where the goal was to protect some part of the lattice from an infection that started in the top right part of the lattice. What we observed is that the neural ODE controller was able to protect nodes more efficiently, and I think at a slightly higher cost, but more efficiently in terms of a reduced overall peak of infection. Now that's something that I wanted to add for discussion. So I think some of the examples that we looked at were more inspired by the question that we wanted to understand the main advantages of these types of neural ODE controllers. But I would also like to, in the future, apply and use these methods to solve some problems that are more applied or more relevant in the medical literature, for example. And one topic that I'm interested in, and maybe some of you are interested in it too, is a treatment protocol optimization. And for example, in the context of antimicrobial or antibiotic resistance, one is often confronted with different types of drugs that one can combine or not combine, or cycle, right? Like there are all sorts of different treatment protocols that one could implement. And one of the challenges is that different protocols or different types of drug combinations can be very hard to optimize because the optimization space is getting very large. And there's a paper that was published a few years ago on operations research on optimal control of an infectious disease with applications to antibiotic resistance. and they used standard traditional control theory approaches. And they were basically, I mean, it's a great paper, and I really like the results, but you will see that it's very hard to optimize these problems. And they were basically just able to focus on a single truck. So they were just optimizing the single truck case. And I think it would be also interesting to focus on larger scale problems. And that's one of the figures that they added. So there are some no treatment regimes and some regimes where one should treat all patients. And these two phases or these two regimes depend on the initial size of the infected population and the initial truck quality. And I think that these findings are very nice, and still there might be some interesting applications of these methods to truck or treatment protocol optimization. And to summarize, so neural network control approaches are useful to control high-dimensional dynamical systems. When compared to standard adjoint gradient solvers, we observed that the methods we use, these neural ODE methods, are up to 2 orders of magnitude faster. And we also applied them to stochastic dynamics, and it worked quite well. And of course, there are various applications and possibilities for future research. One could also think of systems that are only partially observable, or one could also think of on some type of combination of neural network controllers and neural network-based modeling tools that one can use to model systems that are hard to describe with standard methods. Right, like our codes are available on GitHub and on GitLab. You can find references in some of these papers, and also here, like that's the GitHub link. And yeah, I will stop here, and I hope that we can discuss some of these points after the second presentation. Thanks a lot.
Great, thank you. That's intriguing, and I hope there'll be lots of questions. I have a lot, so definitely.
Are we going to have the questions now or at the end?
No, we're going to hold them if it's okay, Gary.
All right.
So our second talk will be from David Gibbs, who's going to be talking about patient-specific cell-cell networks in disease progression. And I don't want to take up your time, so I will turn it over to you directly.
Okay. Okay, let me share my screen. Okay, there. You can see my slides.
Yes.
Yes. Okay, thank you. Okay. Thank you very much for asking us to give a presentation on this material today. My name is Dave. I'm a senior research scientist at the Institute for Systems Biology in Seattle. And this will be a little bit more maybe casual, but I thought I'd start off with a little bit of history of how we got here. And way back in 2017, we were working on a multi-scale model of pancreatic cancer with Boris and Ilya, who now are working on digital twin models with Paul Macklin, who you may know. And as part of the pancreatic model, that's a 3D model of cells that are interacting. And that's when we started looking at kind of how cell communication is happening in cancer. At the same time, we were working on the TCGA pan-cancer paper that was looking at immune response across 32 different types of cancer and about 10,000 samples. So in that work, we were also starting to kind of do statistical analysis of looking at how cells connect to each other, and how that might change in different tissues and different subtypes of cancer. And that work actually went on to be part of a CRI iAtlas, which is a nice web resource that's available to everyone. And I encourage you to check it out. And so kind of through that work on the pancreatic model, kind of this question came up that I became really interested in, which is how, whether you could kind of see patterns of cell-cell communication changing with progression of disease or different phenotypes across patients. And what I really wanted to do was build patient-specific networks. So you'd have a network of cell interactions for each patient that then you could kind of look across and compare and do kind of analysis on that level. And so that was kind of where we were trying to go. One of the papers that was really inspiring to us was this one, also from 2017. It was Friedman and Gallon and others. And you can see kind of on the left that the tumor microenvironment is really a mixture of a lot of different cell types that are all interacting. So when you look in a tumor, it's not just cancer cells. There's really a lot of activity going on there. And these cell interactions are kind of mediated through signaling molecules. So on the right-hand side, you'll see that there's a number of cell types at the top. They're emitting different kinds of molecules. Those are ligands, I call them ligands. And those are then binding to cell surface receptors in that bottom row of cells. And there's a lot of kind of promiscuity that takes place where, you know, one particular ligand can bind multiple receptors, or, you know, one receptor can bind multiple ligands. So it's quite, maybe you'd say a multiplexed kind of network. We also found around this time there were starting to be kind of databases building up that were containing a lot of these ligand receptor interactions. So it was collections, and a lot of times you would have some degree of evidence behind it where you could say, okay, this ligand definitely binds to this receptor. And so these are these really nice resources becoming available that I thought, okay, you know, what can we do with this? And we're also working in cell deconvolution, because in TCGA, those are gene expression profiles from bulk tumor samples, which means that each sample is containing a mixture of a lot of different cell types. And so these methods aim to kind of try to estimate what types of cells were in that sample and the quantity of those cell types. This particular method is more akin to kind of a gene set scoring family of methods. It's called Excel, and the convenient thing is that they provide results for all of the 10,000 TCGA samples, where they have estimates for 64 different cell types. So that's fairly useful. However, if you go to Excel and you're looking at the cells there, and then you have your phantom network scaffold over there, well, there was a real disconnect between these two resources. So we needed to kind of build that bridge between them. In order to do that, we went back to the primary data that they used in that Excel work, where there's a number of kind of large scale experiments where they were sorting out a particular cell. So say CD8 T cells, they sort that out. They have a purified cell type, and then they generate some gene expression profile for it. So you can look at, okay, let's see CD8 T cells, what ligands, what interleukins are they expressing, or what receptors are they expressing? And that gives us a way to connect these cells to that phantom ligand receptor network. But when you start working kind of in this domain, one of the things that is can be pretty painful is that there's a lot of variability in naming cells. And so that was a big effort, was to just kind of unify the cell names. I would encourage everyone to use more like ontologies, like the cell ontology has a pretty good set of cell names that are agreed upon. And if we all use the same naming convention, it makes our lives much easier. So we could start building a network. This network essentially connects all those cells in Excel through that phantom database of ligand receptors to cells. So you have kind of cells emitting ligands, which bind on receptors, and those receptors are on particular cells. It ended up being about 1 million cell-to-cell paths. So here's kind of the basic unit. We have cell L on the left, which produces some ligand, which binds to a receptor, which is on a cell. Now, what I really wanted was some kind of probability statement here. And so you can see kind of below this edge, what it ended up being is kind of the product of two joint probabilities. On the left, we have the joint probability of this cell producing this ligand. And then on the right, we have the probability of a cell having a particular receptor. And we just take the product of those. Now the joint probability can be decomposed into this kind of larger statement on the bottom, which then it's a little easier to deal with actually, where you have the probability of cells and you have probability of ligands given cells, and likewise with the receptors. Now, this statement actually gets used as an edge weight. So for this edge here, we'll make this calculation and we'll use that as a weight and we'll put it on the edge. And we're going to make a big weighted edge, big weighted network for each patient. Now, I was going to do this. It wasn't actually 10,000 samples. It was more like about 9,000. But in order to kind of make these calculations, we needed to generate a lot of distributions ahead of time. So first, we produced the distributions for each cell type. So in this case, we're looking at, say, a CD8 T cell. And these are just empirical distributions, but they're over all of the samples. And then likewise, we have distributions of a particular gene expression given a cell. Okay, so we have kind of all these distributions set up ahead of time that we can use. So here's just to kind of walk through an example of the calculation. So above here in portion A, we might have one edge where we have a CD8 T cell producing IL-2, which binds to the receptor, which is on an NK cell. Now, for one sample, we would come in and we have the CD8 T cell level for that sample. So we get that distribution and we kind of integrate on it, find, okay, that's good, have that. And then given that we have a CD8 T level, here, I broke that into quartiles. And so we look and say, okay, it's a high level of CD8 T cells. So we get that distribution for gene expression and integrate that level for this sample. And below here, we can see this first portion of the statement. We're essentially looking it up using the distributions, and we make that product. And then likewise for the other side, the receptors, and that gives you a weight. The goal, what I really wanted was a big matrix like this, where the rows are going to be each sample. So that'll be a tumor sample in each case. And the columns here are going to be particular cell-to-cell paths. And it's a very large matrix. But once you have that, you can kind of start doing statistical analysis or, start having fun. So one of the first things we saw was that you can identify tissues. So here, for example, this is skin cancer or melanoma, and you can find an edge that's very high in skin cancer and very low in, say, breast cancer, which is in purple here. Or over here, this is pancreatic cancer. You can find edges that are very high just in pancreatic cancer, not really anywhere else. This is lung cancer here. This is a squamous cell. Next to it is adenocarcinoma, lung cancer, and head and neck is here. So interesting, that's also kind of between these two. And then over here, this is stomach adenocarcinoma. And the other colors are from other GI tract cancers. So here in blue, we have rectal cancer, and then colon cancer in purple, and esophageal cancer in orange. So kind of nice to see some similarities across kind of like related tissues. And then I will just go through some of the results here briefly, and then I'll be done. So one of the first results was that indeed you can kind of take if you're taking link by link, so we're just looking at kind of cell to cell link, you could certainly kind of statistically associate those with phenotypes. And we were particularly comparing short versus long progression-free intervals. And a progression-free interval is the length of time when your cancer is not progressing. So in this case, a long interval is kind of the desired outcome. And then we were also looking at early versus late stage cancers, and that can kind of be used as a proxy for kind of how far along the development of the disease is. And so in kind of all of those cases, you can find edges, which here are a thin column of these heat maps. That'll be one edge. And you can see that these are the median edge weights between the two groups. And so indeed, you can find pretty big differences by phenotype. And then once you take those differences, they're kind of, I was calling them differentially weighted edges. And you look at them on that scaffold, you can find that they form connected networks. So in this case, here we're looking at, these are the results from melanoma with the progression-free interval test. And you can see that here, this is what's called a biofabric. And so here, each horizontal row is a node in the network. And if there's a connection between nodes, then it's a vertical line. And so we could find there were edges that kind of associated with short PFI and edges that associated with long PFI. But the interesting thing is that it's a connected network between cells. And you can take the genes that are, found between these cells as making the edges. And you can do things like gene ontology enrichment or gene set scoring. And you can find kind of results that seem to make sense. So for instance, in the stomach cancer, you might see cell-cell adhesion changes. Well, that could go along with the development of metaplasia or dysplasia in the tumor, where the cells kind of start becoming disordered and they're not connected to each other as well anymore. So that's fine. And then lastly, you can use predictive models. You can train them on this data to predict phenotype. And then you can go in and say, okay, well, what features, which would be edges here, which edges are most informative for making a prediction on some phenotype? And you can get kind of a smaller network, which is, very much useful for predicting the phenotype. In this case, I'm using XGBoost models, and that seemed to work pretty well. So we have a lot of networks, and all of this data and scripts and material are available in my GitHub account, which is in the paper. It's pretty cool. We could find that cell edges were, could be associated with different phenotypes, that was a nice result. And the other cool thing is a lot of this material is in Google BigQuery tables, and actually calculated the statistics were kind of a difference of medians. And these statistics were all actually calculated in BigQuery, which is kind of cool. So there's some problems for sure. Some really common links that you expect to find in cancer, that if you're looking at the cancer literature and find an interesting cell-cell interaction, sometimes they're not present in those big databases of ligand receptor. So there's definitely things missing. And secondly, I didn't have cancer cells in the deconvolution results. It's really difficult to try to estimate how many cancer cells are in a bulk RNA-seq sample. That to my understanding is kind of a not solved problem. I wasn't really using the network structure as training on structure necessarily. I was kind of training on edges and then observing networks. So someday I would really like to kind of use the structure itself. And when you're looking across edges, there's a lot of correlation in there. So it's kind of hard to tell, whether that's biological or technical. And technical artifacts, there's sort of a problem with self loops where it's like one cell that's talking to itself. I mean, other cells in the tissue, but sort of they seem to be kind of amplified in the results. And lastly, this is very within TCGA. So there's definitely needs to be some thought on how you could train something on TCGA and go to another data set and have it be useful or combine data. So that's it for me. I'd like to thank my lab. The Shmulevich lab is really great to work in here. And I also work with the Huang lab here. I work with CRI IATLAS, and I'm working on the CRUK is also supporting me, along with HTAN and the ISB CGC. So thank you very much for having me.
Okay, great. Well, thank you to both of our speakers for covering complex topics quickly. Of course, that leaves room for a lot of questions. So the floor is open to questions for both of our speakers.
So if I may start. So Lucas, I'm going to ask questions of you first because that was a really interesting talk. So I think my main question is, what form do you need to have your target system in terms of representation with equations, et cetera, to be able to use this particular method?
Right.
So you say that you have stochastics. I'm assuming those are stochastic differential equations. So the question is, what is the representation format that the target system needs to be in so it can be interfaced with the neural ODE?
Mm-hmm. So one can just start with simple ODE models and basically just need some standard ODE type description of the underlying dynamical system. And then you need a numerical solver, but that is already provided by many of these libraries that we use. So basically you just need a function, something like right-hand side of that depends on some variables in the deterministic case. And in the stochastic case, so we've simulated discrete time, or we've controlled discrete time stochastic dynamics. And the only difference there is that you need to work with like sometimes relatively large batch sizes so that one has enough information about the underlying stochasticity. But sometimes, and that was a bit weird, it also worked extremely well when you just used one sample, which is sometimes in other applications, people call it like one-shot learning. So you just optimize on one sample and you optimize it very well. And then it generalized also quite well. So like again, just to summarize, but what you basically need is just the right-hand side of your dynamical system. And for stochastic systems, the training is a bit more challenging if you want to control it. Yeah.
Yeah, so that's kind of what I was thinking of, because Reinhardt and I had participated in an MBIOS workshop on trying to apply optimal control strategies to agent-based models, which are very useful in representing biological phenomenon. And one of the things that came out of that particular workshop is that this fidelity of representing the biological features of interest is really, really well suited to agent-based models, but none of the control approaches could be used on the agent-based models themselves. You had to have some sort of transform into an equivalent differential equation form so that those particular methods would be useful. So if I can interpret kind of what you've done is you're using the neural ODE training process is essentially identifying the control equation from a complex dynamical system that's represented as a differential equation.
Right. Okay. Is it possible for the agent-based models? Like in some cases, it's possible, right? If it's deterministic, you can have some kind of automata dynamics, some kind of matrix vector multiplications, probably.
Yeah. So I think maybe the spatial disease progression model, I couldn't tell exactly, but if you have like a two-dimensional cellular automata that's deterministic, right? I can imagine that being doable because the underlying dynamics of that system can be readily extracted in equation form, right? So I think.
But you could also, so if you're interested in some of these applications, we've done the same with a colleague from Rotterdam, and he asked us if we can help him with optimizing supply chain dynamics. And that was also a bit special because the dynamics were quite different from the systems that we studied before. And if you're interested in like a specific system, you can also send me an email and I can look at it. Like we basically need we need to differentiate these systems. So there needs to be some kind of so some properties need to be fulfilled so that we can represent some of the control dynamics by a neural network. But I can check it if you send me
Yeah, I'll send you an email
Yeah.
because what we do is we do the DRL on the simulation model itself,
Okay.
But I'm curious, right? Because the question is, it's pretty computationally intensive.
Yeah.
But the precondition necessary for this is that's what I'm curious about. So I'll send you an email and let you know.
Perfect. Thank you.
So if I'm going to still talk, all right. So David, I have a question for you with respect to this. You addressed part of it having to do with this perpetual question about when you take a tumor sample, how much of it is actually cancer and how much of it is actually stromal tissue, which is a perpetual problem associated with this. So one of the questions I had to do with the dimensionality problem with respect to overfitting, because you're dealing with all these, genes, et cetera, and even leaving aside for a second that gene expression doesn't equate to function, so the presence of that gene doesn't necessarily mean that the ligand is being produced or the receptor is being produced. That correlation matrix, it's going to fit. You're going to find a fit, right? Because you have so many parameters. So I guess this may have to do with the question of how you are going to expand this. So what do you think about that?
Yeah, you're certainly right. There's a lot of features to try in making a prediction. I guess it was kind of nice though to see that you end up with functional enrichments that make sense for the particular phenotype you're looking at. So I guess you can kind of like go back afterwards and see if it makes sense.
Yeah, there's a sanity check, a plausibility check, in retrospect.
Mm-hmm. And certainly, I think, yeah, I don't know for sure.
All right.
But,
it's difficult, right? That's why you're still working on it.
I think that moving towards single cell data is going to be really useful. And actually, it's a better fit for these kind of methods because you're not even having to estimate what cell is there and if it's producing a ligand or not. And even there's single cell methods that measure transcripts, but also measure surface proteins called site-seq. And it does it simultaneously. So then you actually get even a protein measure, which is what you really want. So I think maybe moving to some newer single cell data could be useful.
Thanks. And I'm also going to assume that part of your involvement with Paul is that you're going to convert these kind of networks into directed graphs and instantiate them in PhysiCell as rules for an ABM, right?
I'm actually not working with that group. My colleague, Boris Aguilar, is, and he would be a great candidate to come and give you a talk on that, actually.
Yeah, I think that's a natural extension of this.
Yeah, I agree. And that's where I guess also you could test if you're making good predictions, is if you can build then a model,
right.
like a
mechanistic dynamical model.
See if it actually makes sense.
Yeah, because then you can perturb that model and theoretically determine whether or not you have a plausibility check in its perturbation response.
Yeah, and maybe using a system where they've done drug treatment on like a dish of cells. Where it's a real controlled environment and you kind of know biologically what should have happened and you can see if you can kind of computationally reproduce that. That might work.
Yeah. I think organoids might be an interesting in vitro system to do this, right? Where you do this kind of network reconstruction from a constrained experimental platform that you can then perturb that platform as a representation. And, so you do your network analysis, you do that reconstruction, you use those reconstructions to construct a simulation model. You have a simulation model that makes predictions as to what a perturbation looks like, and then you test those perturbations in an organoid model. That would be interesting.
Absolutely.
I would love to work with you if you are doing anything like that.
That's a good idea, because I know some labs that are producing organoids right now through our CRUK work. So they're out there.
And they're looking for means of encapsulating the knowledge that's present in that, because the organoids themselves, since they have that extra layer of complexity beyond standard cell culture in vitro stuff, there are challenges with respect to the inference as to what comes out of those systems.
That's true. Yeah, like there's a difference between the inside and the outside, right? So there's like orientation.
There's a spatial heterogeneity associated with it.
Yeah.
I mean, you can get the inside outside with a monolayer culture with the luminal side and the apical side, et cetera, but or the basal side. But there's the multicellularity the multi-cell type possibilities of organoids, I think, are important.
Mm-hmm.
But very nice.
Other questions, comments? I mean, I guess coming back to what Gary was talking about, and we heard about this last week in more detail, which is, I mean, the Go term associations tell you only in the most vague way what things do. They don't even tell you the direction of the effect. They'd be told that you see enhancement in genes associated with cell motility or cell proliferation in metastatic cancer. Doesn't give you a lot. I was just working with a student today. We were looking at that omics paper, and everything that comes up, I know. And not only do I know it better than the paper does, because the term annotations aren't really very informative very often. It has something to do with metabolism. Great. Now, of course, the linkage between receptors and ligands is a much more biologically meaningful set of results. And that certainly has a spatial context. So the kind of modeling Gary and I do, agent-based models where you put networks in, but it still does leave the question, which is exactly what Gary brought up, which is how do you take a network and instantiate it in a way that could control the cells that you're simulating? And that's not trivial to do. Gary uses rule-based methods. We tend to use more dynamical network methods, which are heavier and maybe don't give you a lot of additional information. But I think that there is an interesting question about how you take the kind of really interesting qualitative information that you're producing here and connect it to the kind of mechanistic modeling that Gary and I and Paul do, or Reinhardt. And so I don't know if you have answers for that. I agree with Gary. I'd love to talk about it. And I think last week we saw a little bit of actually trying to back out specific GRNs from this kind of information or specific metabolic connections.
Yeah, the problem is that those have to be experimentally identified so that the detailed information is possible. I think it's impossible to make an inference about those sorts of things, from the aggregate data. You just can't do it. You don't have enough samples to be able to make that inference reliably. So I think the main pathway of these gene regulatory network identifications or structures or whatever are to identify motifs that are of particular interest that then can be interrogated, experimentally for more detail. and I think the other issue here is, and this is I think an interesting question that we can talk about at some other point, is gene expression the appropriate resolution for if we want to apply pharmacologic control? Because as we talked about, gene expression does not equate to protein, right? And protein is a functional form by which cells communicate with each other. So, I mean, essentially the products, whether you're talking about, excreted RNAs or whatever, it's some product of the gene expression that isn't necessarily a direct relationship between the expression of the, of the RNA initially. So, so I think that there is a quagmire inside the cytoplasm that is really, really, really complicated. Now, in certain disease states where you're looking at oncogenesis, right, and disorders of the regulatory mechanisms of the cells, I think it may be important to look at that. But with the increased interest in tumor stromal interactions as the thing that may actually drive tumor behavior, as opposed to the specific properties of the cancerous cells per se, I think this idea of thinking of differential cell populations interacting with each other represents probably the resolution for therapeutic targets, as opposed to trying to control gene regulatory networks per se.
Yeah, it reminds me there's one way that people are trying to ascertain whether the cell-cell interaction has some kind of effect taking place, which is then if you know what's downstream of the receptor, ultimately receptor all the way to the regulatory aspect. And if you can observe a set of genes, a gene set that is then upregulated or downregulated from that receptor, then you can start kind of inferring like, okay, maybe this cell-cell connection is having an impact in the cell. So that's
also, but you need to actually take that to the next step, because that needs to result in some change in the functional phenotype of that cell,
yeah,
right? Be it either secretion of protein, change of state, or whatever.
Yeah.
Because the same problem exists once you get to that particular constellation of intracellular genes, whether or not that actually is representative as a functional phenotype. So this is part of the problem associated with all the cell receptor oriented classification of immune cells, right? Because there's like a bajillion different types of subtypes of T cells and B cells, et cetera, and they're characterized by all these receptors for which no one has any idea what they actually do. They're just different, right? And it's unclear what the functional differences are between all these different types of cell types, except that they express different cell receptors. So I think, that there's a natural reductionist tendency to want to be able to classify things at this scale of resolution, but at some point in time, right, it is the functional consequences of that, given whatever disease process you're looking for, that is, what the actionable piece of information happens to be.
I had one or two. One is a comment more than everything. So with your method of essentially untangling the cell type within out of bulk RNA-seq, I guess the answer is no, but it might be I might be wrong. Can you appreciate somehow the heterogeneity within cancer cells, which is known to be kind of a problem, notable problem?
No.
Okay.
Not really with this particular method, because that's still missing, is the cancer cell itself is missing, which is kind of a big omission.
The other that is just for discussion, I guess, is I think it will take a lot of work, but I think something very interesting will be to couple spatial and information that you get from imaging, which is a thing that I'm very interested in, even eye contact, etc., with this genomic information. And I think the hard working part will be probably doing some laser capture microdissection, where you pick the things and perhaps in serial section of tissues, but that I think will add a lot of baseline information to the efforts of simulating, etc. And then also probably doing with healthy tissue, because we always when we think cancer, we always concentrate on the cancer, but then the healthy tissue has, I think, a wealth of information to give us.
Also spatial transcriptomics, is part of this too.
Right.
Yeah.
And we're working with HTAN. We're part of the Data Coordinating Center. And if you keep an eye on that, that's the Human Tumor Atlas Network. Very soon, people are going to be starting to make a lot of spatial transcriptomic data public. And it's in a number of different levels, from raw to more and more processed, and to, something that we can work with that is, cell identifications, and you have, location, and then you have a number of protein measures on each cell. It's very cool. And I do think that is going to be really interesting to see how things vary spatially. It's really just scratching the surface at this point. And yes, that's it. That's exciting stuff. I'm really excited by that. Here at ISB, we also have a new machine from NanoString that is called the Geomics. And it, instead of kind of like cell level, it takes a region of interest, which is a small patch of cells, and then can measure protein and gene expression levels in that little region of interest. And then you take a big slide and you pick out a bunch of regions. So those are kind of your spatial measurement there. And I haven't seen the data yet, but we're working with the lab here that's just starting to produce it. So I'm pretty excited to see that. Yeah, that's coming next for sure.
Lorenzo, were you done, or other questions from the audience? I wanted to come back, Lucas, because you went through this quickly. Gary has been doing this for a long time. So just to understand, what is the fundamental loss function you're training on? Is it just the distance from the final point of your dynamical system to the target point, or are there other terms of the loss function as well?
Right. So for most applications that we studied in the time-dependent control paper, it's just the distance from the target state. And as I said, like usually if you do standard PMP Pontryagin optimization on a like pencil and paper calculation, then you also account for some integrated costs. And we didn't do that in many applications, and it still produced very low energy results. But of course, if you consider other applications, I think for the so in feedback control, when you at the state of the system into the loss function, you have to do some more engineering to come up with a good loss. But otherwise, if you just want to reach a state, MSE distance between initial and target, or reached and target, worked really well. Yeah.
Target end state. Target end state.
Yeah, right.
And so let me understand, because that doesn't sound like it intrinsically requires you to know the moment by moment dynamics, because you run the simulation, you see what the final state is, and you then have it returns a loss. So you don't need to have a differentiable system to do that.
Well, it has to have a trajectory that it fits to as it goes towards that path. Right.
But that's what I was trying to understand. But if the loss function doesn't include the trajectory, It
So,
is.
so the thing is, um, and that's also something that I got really confused about for almost 2 years. So if you and I'm about to summarize it and maybe have a more technical paper on that. So the thing that is happening is that your loss function just depends on the reach and target state. But if you do these calculations, you basically have an integral term in your loss function. So you integrate over something, you have all these steps in between. And on your computer, you basically have a computational graph where you have all these control inputs that affect every single step, and they depend on your neural network weight. So there is some so there's some transform that is basically making these gradient updates a bit more complicated. Yeah.
So I guess I missed what it is, how you actually, what is the control you're applying to your system? So I have a dynamical system. The dynamical system has a state,
Yeah.
and it has a set of parameters. I put that in and it gives me a time evolution.
Right.
And I could control it by nudging the values of my variables.
Mm-hmm.
I could control it by having the parameters be time dependent.
Mm-hmm.
What are the controls that I'm allowed in or that you use
Could
to do
be
this control?
could be anything. So you could have something in the form of x dot equals f of x, and maybe some parameters, right? Maybe some alpha and beta,
Right.
and it could be, so the control could be multiplicative. So you could have a multiplicative time dependent function in front of your state. It could be modifying other variables. So it could be any type of time dependent control function that affects your F, the right hand side of your ODE.
But do I apply it? But I could, so I write that I can't control everything. You attach the control to particular parameters or particular variables.
Mm-hmm.
Right, you
And
can, I think, specify the control interaction space, right, by how you set up the interaction between the neural ODE and the control target system, right? Just as you would with a control space, if you're doing RL. But I think this integrating component is the key here, is because you need to have either a pre-specified or readily extractable trajectory space to get from point A to whatever your control target has to be, right? Because without that particular function, there's nothing that it's going to integrate over, because if it's everything, it's everything, right? So, right. So when you do RL on things like this, right, if you try and do RL only on end state, it will take forever, because you don't know what the individual control functions are going to be, right? So you have to have, intermediate cost functions, right? Not end state cost functions as surrogates for what you're integrating over if you're going to solve it with a known dynamical system, right? Because you need to have some directionality of where it's going to go next to be able to integrate towards that particular ideal control trajectory, right? With the neural ODE.
So that's for the neural ODE part. But I was also thinking about the system that you mentioned, and we did something similar by just looking at so we studied other systems where we didn't really, so we just took an end state that, like it was another type of system. So we had a system where we accumulated some cost over time, but it was not differentiable, just some like scalar value basically, and we just tried, and that was for the stochastic system, and we just tried to minimize it, and we and we basically just parameterize the control function for discrete dynamical system also by a neural network, and we did some optimization over large batches, and that worked also quite well. That's why I like for the system that you described, I think I would probably try something like that, like some direct optimization, not involving any neural ODEs, because I think for the agent-based models that you mentioned, it could also work,
yeah.
but I have to see. Yeah.
Yeah, so you need essentially a time series of cost functions, right?
Mm-hmm.
That you step over. Yeah, I think that. So I think identifying the at least the challenge that we have in our, very stochastic, very complicated agent-based models is that we can't necessarily make any sort of inference about what the multidimensional generative state space is with respect to our system level target function without simulating it over and over and over again. We can't, right? We can't actually extract what that looks like without running it, millions of times, because it is very stochastic. And it's
but you know what you want to optimize, right? So you have some.
Well, we have an end state that we want.
Okay.
Right. So for instance, we have, just to talk about it in general, it's let's call it an in silico analog of a person. All right. And you perturb the person and the dynamics of the system actually runs, and it either dies by some criteria or it lives. We want to make it live, right? That live is an aggregate output of all the entire microstates of the 50, rules and parameters and mediators or whatever that's represented in that particular system. Those things are all interacting with each other. They've got a bunch of stochastics associated with that. You can conceive that there is a path to that particular point, but that path is not unique in any straight way or way, shape, or form. And it is highly variable, right? Where there are basins of stochasticity where, so it's a very large set of chaotic attractors. Let's describe it in that particular fashion. So it's not deterministic, which means that unless you get into one basin for sure, it becomes deterministic at some point in time, right? Where the system states irrevocably fall to one attractor or another attractor. But there's a huge region of undecidability in the middle, right? And if, depending on how you want to apply your control, it is theoretically possible to shift from a deterministic, probabilistic basin out of that basin into another control space, right? Into another control trajectory. So thinking about how to represent in let's call it a compressible form, right? So that you can integrate over what that trajectory looks like. I just have a great deal of difficulty visualizing this.
Yeah. I have to look at it, but like we tried out many different systems, and we also adapted our methods all the time. So what I call neural ODE control is basically just one paper for the time dependent setting, and there are different papers for different types of control problems, because you always have to change the method a bit. So, but
I'll send you an email. We can correspond about this a little bit.
I mean, I guess, Gary, suppose I wrote an ODE tumor model where I had some functional form for the growth of the tumor, a rule which was the fraction of tumor cells that were resistant to my chemotherapy. And that was also some differential equation. And I now gave my chemo, and I allowed my chemo to be given with some maximum dose on any possible time trajectory. And I want to optimize so that I have as few possible resistant cells at the end and as small a tumor as possible for some trajectory of my chemo that's a well-defined dynamical system to begin with, and it should be soluble.
Yeah,
Now,
so
I
I mean,
do the same simulation with agents where I have cancer cells that divide, grow, and so on. I could define a set of agents that are derived from the ODEs that in the limit give us that, although the individual models are stochastic. So if in principle, one ought to be able to solve the same problem, but in practice, solving the individual agent-based models is incredibly time-consuming compared to the ODE solver.
well, so it all starts
It's not
with your
differentiable anymore.
I mean, it starts with your initial assumption that the system you're interested in can be sufficiently represented with a sufficiently complex ordinary differential equation. Because once you have the differential equation model, you can use this method, right? The point of that entire workshop that we went through is that there are sufficiently complex biological systems for which you can't do that. Right. And a lot of those are the ones that we're actually interested in. Now, it doesn't mean that you can't represent a particular subgroup of the behavior of that system with an ODE. Right. But if you're trying to, and again, it goes to what your actual question is. If the question is to try and replicate the heterogeneity of the biological population, right, your ODE is not sufficient to do that. Right. This, to a certain degree, is the whole point behind how we need to think about representing digital twins if we're going to do digital twins for drug development, discovery, and retesting, because they are presupposed on a mechanistic representation of these particular factors that, as the goal of a digital twin is, need to be able to represent anybody. Right. And so if you're going to represent anybody, you have to be able to represent everybody, which means that that system has to be able to capture the entire breadth of biological possibility of that particular system. Right? Doesn't mean that you can't apply it to a particular, right? So here's an example. So the cancer chemotherapy optimization strategy is not an unreasonable thing to do, because you have a group of patients of which a particular piece of biology you're interested in. You can create a semi-predictive model of what the differential responsivenesses are of known chemotherapeutic agents for that particular cohort of patients. And as long as your patients falls within some of the classes that are present in that particular optimization strategy, it will work. The problem, of course, is that there will be patients that do not fall into those particular areas, of which this will not tell you anything about what to do about those particular patients, right? So there's an optimization task, which is perfectly fine, right? But the optimization across the entire space of available therapies is not sufficient to find treatments for the people that fall outside that particular category, which is what we actually want to do, right? So they're both useful tasks, right? But the tools need to suit what that task happens to be.
Gary, I wasn't trying to propose that the ODE system was in any way a complete explanation. I was just trying to give, and maybe it was a bad choice because it was a tendentious choice of example. I was just trying to think through the workflow of going from the ODE, which we heard about, to trying to extend that optimization problem to the agent-based analog. I wasn't trying to say that the ODE system was a sufficient explanation of any kind. I was just trying to think of some example where you could build a pair of ODE system and agent-based model, potentially apply the optimizer to the ODE system, and then see how that optimization played out in the ODE model. And maybe use that
in the agent-based model.
Inside the agent-based model, yes.
And we can really try. So I also have a few collaborators who are always interested in trying out new things. So we can really try to optimize some of these agent-based models directly, even if it looks complicated. So these stochastic systems that we optimize for the colleague in Rotterdam, that system can be solved analytically, and they usually use some dynamic programming optimization approaches for very small scale problems that took, like, on my machine, it took 50 days to find a solution that was as close as the solution that we found with not the neural ODE optimizer, but with another neural network after 30 seconds of transfer learning from another solution that maybe took 5 minutes to find. And that's also like a very high dimensional problem in terms of action space. So you have too many actions to optimize these problems in a reasonable way. But somehow I think if you do like gradient descent optimization over very large batches, there seems to be some efficiency gain, so that you converge towards minima that are close to optimal. Yeah. So we can.
Yeah.
I would try it out. It sounds really interesting.
Yeah, we'll definitely be in contact. So James, what you described is exactly what Reinhardt tries to do, right, as a byproduct of that BMB paper, right, is to identify sufficient, ODE surrogates to do control on them and see how it works. So.
Okay.
Well, another time, I guess. Yeah.
I think we're out of time. Lorenzo, did you have a question? Or
I think it's a little bit long.
Okay.
It was the inverse question on if we are able to infer some function, some surrogate function of our system in a, let's say, limited system with few parameters, would that solution, would that function obtained through the neural network encapsulate the biology or the nature of the system itself, or it will be just a numerical optimization? And I guess this is for another time.
Yeah, so keywords are also physics-informed neural networks, I guess. So there are some applications to biology where you specify some parts of the dynamics, some constraints that are physical or biological constraints. And the other keyword is also basically just neural ODEs. You basically know that x dot equals some f of x, and you try to infer the f of x based on observations. And I can also, I have your email. I send you one of the notebooks that I use in my lecture, and it's exactly that, like how to infer a system from observations. So I send it to you.
Thank you very much.
Okay, great. Well, we had a lot of comments and questions today. This was great.
Thank you very much, guys.
Thank you, everybody, for joining us today.
Thanks a lot.
We really appreciate the two talks and the discussion. Thank you again. Bye.
Thanks. Have a good afternoon.
Bye.